midterm number 5 coming up next Monday. And I guess I told you the labs that are going to be on it. I shouldn't say the labs, the lectures and the labs that are going to be on it. If you're not sure, talk to me about it all. I'll tell you about it. This Friday here in the lecture hall is your usual old Friday quiz, and that's going to be on this chapter on bacteria and viruses that we've been on up through and including what we cover in today's lecture. What else can I tell you? Oh, today is in laboratory as part of the pre-lab lecture. I'm going to be covering the first part of the DNA technology lecture handout, so please bring that with you to take notes while I lecture on that. All right, well, I guess that's all my announcements. Let's jump back into where we were in the lecture. Here we go. We're talking about bacteria, and the section that we started on here, and in fact the rest of this lecture, is all about bacteria and their genes and how they express those genes. And remember, when we say gene expression, it just means everything from going to the gene to the final protein. Gene expression includes RNA polymerase making an mRNA from the gene, and then mRNA then binding to a ribosome, and then the ribosome translating it into a protein, and then having that active enzyme, that protein in the cell. So to begin with, almost all the genes in a bacteria are on its chromosome, which is double-stranded DNA, just like in our cells. But of course in bacteria, actually in all prokaryotes, bacteria and archaea, the chromosome is circular, you know that. And typically a few thousand genes are on a bacterial chromosome. But remember, bacteria can also have some little circular pieces of DNA called plasmids. Where are you, plasmids? Here you are. And so these are also circular bits of double-stranded DNA. They usually, and they're smaller than the chromosome, so typically they may only carry 100 or fewer genes, and some of the smaller ones I've heard about might only have 3 or 4 genes on them. And the plasmids do not carry genes that are usually necessary for survival of the bacteria. I think I mentioned last time that the genes, for instance, for breaking down glucose and using it for energy would be here on the bacterial main plasmid, since those are genes that the bacteria normally needs for survival. And genes for making the cell wall and the cell membrane, again, you'd find those genes there because those are normal genes for helping the bacteria survive. But genes for things like antibiotic resistance, which is not normally needed by the bacteria to survive because most bacteria are not being exposed to antibiotics at any given time. Anyway, those bacterial resistance genes, for example, might be the ones found on plasmids. I always think of those as bonus genes that might help the bacteria but aren't necessarily needed for it to survive under normal conditions. And I might have mentioned this, that molecular biologists have found ways to use plasmids to help express human genes. They can take a human gene and attach it to a plasmid and then add that plasmid back inside bacteria, and then the bacteria becomes a factory for expressing that human gene. All right, good stuff. This you've seen before, bacteria reproduce by a process called binary fission, which is a very rapid way of reproducing. I think this is where we left off here. When bacteria reproduce by binary fission, that's not sexual reproduction. The daughter cells are exact clones of the mother cell, all the same genes. Bacteria really do not have sexual reproduction. There is no male bacteria and female bacteria mating with each other. It just doesn't happen. No such thing as male and female bacteria. And yet, they have shown experimentally that one bacteria can pass genes to another bacteria. This is a diagram of an experiment that they did. They took two different species of bacteria, this one here and that one there, and one species had a functional gene. And if I remember right, the gene was for making a certain amino acid. You know, all cells need amino acids to survive, and so they need genes for manufacturing those amino acids. So here's the functional copy of that amino acid making gene. And this other species of bacteria, they gave a non-functional copy of that gene. And they mixed these two bacteria together. They let them grow in the same flask. And what they eventually found is this top species of bacteria here, some of them at least, got that gene somehow from the other bacteria, like that. They ended up getting the good gene in their chromosome. And you know, if bacteria did have sexual reproduction, this would be easy to explain. You'd figure these two bacteria mated with each other, and so when two things mate, they both contribute genes to the offspring. So you might think at first, well, this is an offspring bacteria of the two original ones. But that turned out not to be the case. It's not sexual reproduction. It's simply this, that bacteria have ways of transferring some of their genes physically, moving the DNA across to another bacteria. And so that new bacteria will then start expressing the genes on the piece of DNA it got. It would be like this. I have genes for blue eyes, and if I went up to one of you with brown eyes and took some DNA out of my body and threw it into your body, and suddenly your eyes turn blue. You know, it's not sexual reproduction, but we're passing a gene between each other. And so they began studying how bacteria can do this, and it turned out that there were actually 3 different methods that bacteria have of gene transfer. And they're called transformation, transduction, and conjugation. And of those 3, the most straightforward is transformation, because it's basically just exactly that, a piece of DNA moving from one or going into a bacteria, like that. And here's a way to look at it. Here's a bacteria, and imagine that drifting around outside the bacteria somewhere is what I'll call a foreign, naked piece of DNA. And by naked, I mean not enclosed in anything. It's not inside a nucleus. It's not a viral chromosome wrapped in a capsid. It's just a raw piece of DNA with nothing wrapped around it. And it's foreign, meaning it's coming from outside the bacteria. Bacteria can actually just take in foreign pieces of DNA that are floating around outside themselves and splice them into their own chromosome. Think about the bacterial membrane for a second. One of the things I've been telling you about membranes is that they function as barriers to keep the stuff outside a cell outside. And so you may wonder, well, how does that piece of naked foreign DNA get inside the bacteria? Well, it sounds kind of interesting, but bacteria actually have receptor proteins on their cell membranes that can allow pieces of foreign DNA to come inside. And you remember not receptor proteins, I should have said transport proteins that can allow pieces of foreign DNA to come inside. And the reason I think that's kind of interesting is transport proteins have binding sites for the stuff that they're supposed to be transporting. So if bacteria have this, and these transport proteins, and they do, it must be because the bacteria actually want to bring in foreign bits of DNA. Like that. And when I first learned that, I thought, you know, that's interesting. It means that bacteria are kind of gamblers, because they have no idea what genes are encoded on here. Maybe they're optimists. Maybe that's a good way to think about it. Piece of DNA, sure, bring it inside me. I'll start expressing those genes. I mean, the bacteria has no way of knowing if this is viral genes or something potentially dangerous, but the bacteria says, sure, I'll bring it inside, not a problem. And then once that piece of DNA does enter the bacteria, it gets spliced into the bacteria's chromosome, and now the bacteria is going to start expressing those genes. So that's the essence of transformation, just bring in DNA from outside and splice it into the bacteria's chromosome.
Do they think that that's done because the bacteria wants to be more robust?
In some way, in an evolutionary sense, it must benefit the bacteria, and so I don't know what to say other than the fact that perhaps they're gamblers and they're willing to gamble that that might be a beneficial piece of DNA for them. And actually, yeah, I want to write something on the board kind of along those lines. So what you're going to be doing in laboratory, well, for today's lab and the next few labs, is a cloning experiment. And I'll explain more about the cloning when we get into the laboratory. But essentially, it's this. We're going to start off with a piece of plasmid DNA, not inside a bacteria, a piece of plasmid DNA that's been removed from a bacteria. And we're going to put in a non-bacterial gene. You might remember from last bacteria, I said we're going to be cloning a jellyfish gene, something called the green fluorescent protein gene, which comes from jellyfish. And we ligate that, we join that onto a bacterial plasmid. And then we're going to, in today's lab, take some bacteria and we're going to transform the bacteria, which means exactly that. We're going to make them take up this foreign naked piece of DNA, put it into the bacteria like that. And if we do it correctly, then the bacteria are going to start expressing this gene, and we're going to end up with green fluorescent bacteria, so to speak. They don't actually glow in the dark, but they will glow under ultraviolet light. As I just got through telling you, bacteria actually have transport proteins in their membrane to bring in foreign pieces of DNA. But the problem is, it's not a very efficient process. Yeah, the bacteria do have those, but they don't have too many of those transport proteins. And so there are other ways to encourage the bacteria to take in foreign DNA when you're doing it in the laboratory like this. And I wanted to just briefly mention some of those. What I'm going to draw next is the part of the bacteria's membrane. Here are the phospholipids of the bacteria membrane. Who remembers the ionic charge on the phospholipids? Positive or negative? Negative, right. They're phospholipids, and phosphate, they have phosphate functional groups, and phosphates are always negatively charged. So all these guys have negative charges on them. And DNA is the same way. Here's this plasmid that we're trying to put into the bacteria. It's also made out of DNA. And DNA has phosphates as part of its backbone. So it carries a bunch of negative charges. And so notice that since like charges repel each other, that's going to actually tend to keep the DNA from passing through the membrane. What did I tell you we have on membranes to sort of neutralize these charges? Does anybody recall? Something you did when you were trying to well, it relates to when you were trying to get rid of the membranes. Yeah, what kind of ions? Calcium ions, that's right, and magnesium. For most cells, they keep some calcium and magnesium ions around their membranes, because what would happen is the negative charges on these phosphates would tend to push away from each other, and that would tend to loosen up the membrane. And so cells usually have a few calcium and magnesium ions stationed around the membrane to neutralize these negative charges, and that helps the phospholipids hang out a little bit closer together. Well, we're going to use the same trick. We're going to add a bunch of extra calcium and magnesium ions to the mixture. Both, actually, I guess we're just going to use calcium ions, but I guess we could have used magnesium ions also. And so these will neutralize the charges both on the plasmid and on the bacteria's membrane. So there's less ionic repulsion between the two, and that's going to help us get the plasmid through the membrane. And we're also going to do another trick called heat shock. When you heat something up, of course, you increase the molecular motion of it, and that would tend to make the membrane get a little bit looser. And so we're also, in addition to the calcium, going to heat up the bacteria very briefly, and that will also help us get our plasmid inside there. So the point of what I'm saying here is, yes, bacteria do have transport proteins to bring in foreign pieces of DNA, but it's not a very efficient process, and there are various techniques you can use in the laboratory if you want to transform bacteria that make it a lot more efficient. All right, any transformation questions?